All right, today, so we are at the part of structures and functions of macromolecules. So there are four types of macromolecules: proteins, nucleic acids, lipids, and carbohydrates. And so in this class, we mainly focus on proteins and nucleic acids. And we already talked about proteins, which is polymer of what? What's a monomer for proteins? Amino acids, okay. So in the first 2 weeks, we talked about proteins, which is composed of amino acids. And what kind of chemical bonds between amino acids? Yeah, what kind of covalent bonds? Peptide bond, yes. Peptide bond, mainly peptide bond, and we also have a disulfide bond. And there are some non-covalent bonds that stabilize tertiary structures. Hydrogen bond and hydrophobic interactions. And there are two more. There are two more non-covalent bonds. That's what? Van der Waals interactions and salt bridge. Salt bridge is also called electrostatic interactions. So for proteins, the monomer is amino acid, the covalent bond is peptide bond, and we have four more non-covalent bond interactions. And for nucleic acids are polymers of nucleotide. Okay. Nucleic acids is composed by nucleotides. And every nucleotide has 3 parts. Does anyone still remember? It has a sugar backbone. It has a sugar backbone. And it has some base. And a phosphate group. Okay, that's a nucleotide. So there are 4 bases for DNA. What kind of bases? ATCG, okay. The base could be ATCG for DNA or AUCG for RNA. And here this is either H or hydroxyl group, depending on whether we have DNA or RNA. Okay, so you can see that by analogy. Next, we want to know, to form a polymer of nucleic acids, what kind of chemical bonds we need for nucleotide. Okay, and this is base. We have 2 types of base, purine and pyrimidine. And they form nucleotide. And there are 2 types of amino acids that have UV absorbance. Which 2? There are 2 amino acids, tyrosine and tryptophan, because that's the 2 that have aromatic bonds. And well, nucleotides, their base, you can look at their base structure. Basically, all nucleotides, their base structure is basically aromatic rings. So all nucleotides have absorbance and UV wavelengths. And it so happens that for the nucleic acids have absorption at 260 nanometer, while tyrosine and tryptophan has absorption at 280 nanometer. Okay. And last time we talked about if the OD equals to 1 for DNA, that means the DNA concentration is 50 microgram per mil. Okay. And so we have this question, the question to if OD 260 is 0.5, what's the concentration of DNA? How do you calculate that? 0.5 and what? If OD equals to 1, that means the DNA concentration is 50 microgram per mil. If OD is 0.5, how do you do that? You just do a product. Okay. The DNA concentration would be 0.5 times 50, which is 25 microgram per mil. Okay. And so the third question is actually a tricky question. Well, if the OD 260 is 2, what's the DNA concentration? What? Well, if OD is 1, the DNA concentration is 50. If OD is 2, what's the DNA concentration? What? 1? 100? Yeah. Well, in theory, okay, in theory, if OD is 2, then the DNA concentration is 100. But in reality, probably the DNA concentration is higher than 100. Here's why. When we talk about OD, we mean this. OD means it's the incoming light. Okay. Let's see. You have a container with DNA or protein concentrations. You have some incoming light, and after the solution, a lot of the energy in the light is absorbed. So the outgoing light is reduced. And if the intensity of incoming light is I, the outcoming light is just I0. This is I0, this is I. And the ratio, and then you take the ratio and then you take the log. And this is the OD. Okay, this is the OD number. If OD equals to 1, that means, if OD equals to 1, that means the I0 over I is 10, okay? That means if you have 10, if intensity, incoming intensity is 10, then the outgoing intensity is only 1. And so 99% of the light is absorbed in the process. Well, if OD equals to 2, that means incoming over outgoing light is 100. So that means 100 light coming in, and only 1 fraction comes out. So, so here it's 10% is left. Here is only 1% is left. So what happens is this: if OD is too high, above 1, that means too much light is absorbed. Then the light that comes out from the solution is very, very weak, and it's not accurate to measure a weak light. So here's a practical reason so why we have to care about this. It's not. So in theory, if OD is 2, then the DNA concentration is just 100. But in reality, if the OD equals to 2, that means there's too much light getting absorbed in the process. you won't have enough accuracy to measure an OD equals to 2. Because this light is just too dim. Your measurement won't be too accurate. So if OD is equal to 2, or actually anything above 1, what you do is this. If OD equals to 2, what you do is you dilute your sample first. Your DNA is too concentrated. You want to add water or add a buffer to your DNA solution. Make the OD below 1, okay, until your OD is 1. Actually, the OD, when you do such experiment, the OD should always be between 0.1 and 1. If its OD is too low, that means your incoming and outgoing light is basically the same. Then the measurement won't be accurate. If OD is too much, that means the outgoing light is too weak, and the measurement won't be too accurate. So always keep your concentration in the range so that the OD is between 0.1 and 1, and then you can measure that accurately. Okay, so this is about the UV absorbance of nucleotides. And then amino acids are connected by peptide bonds. Then let's see what bond connects nucleotide. And this bond is called 3' to 5' phosphodiester bond. Phosphodiester bond. It has a long name. Let's see what it actually means. So first it's a 3' to 5'. Does anyone know what 3' means? 3'. 3' means this carbon, okay? So in the pentose ring, this is a 1' carbon, this is 2' carbon, this is 3', this is 4', and this is the 5' carbon, okay? And does anyone know why we need a prime? Why we need this? Why we don't say 3 to 5? It's a 3' carbon. That's because in chemistry, okay, if we have a chemical bond that have 2 rings, and the number would be different. Because now we have the structure look like this. We have, in any amino acid, we have at least 2 rings, okay? And in the first ring, and we have to number the atoms in each ring. And in DNA or RNA, the numbering on the base, on the ring, on the base is 1, 2, 3, 4, 5, 6. And the numbering on this carbon ring, on this sugar ring, it's 1', 2', 3', 5'. And if there is a third ring, the numbering would be like 2, 1, double prime, 2 double prime, 3 double prime. This is just a way to distinguish different rings. When we talk about like carbon 1, you need to be clear about, are you talking about carbon 1 on this ring, or is it carbon 1 on this ring? So that's why we have the prime here. Okay. And so 3' means this carbon here. So 3' means this, and 5' means this. This is a 5', this is a 3', okay? Also notice that 5' is not this, okay? There's a carbon here, but this is number 4. 5' is outside of the ring, okay? That's 1. And so it's a phosphodiester. So let's see what's an ester. An ester is formed between an acid and any hydroxyl group. Okay. So here we have a hydroxyl group on the 3' carbon. So if we add the hydroxyl group here, that's an ester. And it's a diester, which is because the phosphate group can form another ester bond. So we can let it to form the ester bond of the next 5' carbon. So it will be like this. This is base. Let's see. Okay. So this is, again, the next nucleotide. So we are now talking about the chemical bond between the two nucleotides. The next nucleotide has a hydroxyl group at 5' carbon. And the first nucleotide has a hydroxyl group on the 3' carbon. And the phosphate group can form ester bond with both hydroxyl group. So this is pretty much called diester bond. And since this is always going from the 3' direction to the 5' direction, so this is called 3' to 5' phosphodiester bond. And of course, this one also has the next nucleotide also, now it's a 3' hydroxyl group is exposed. It can connect to the next 5' hydroxyl group by phosphates. And it goes on and on and on, just like a peptide bond connecting amino acids. Okay. And it goes on and on and on, and you can see that we have the base in each nucleotide could be different. And so we have basically a long letter of ATCGs. And this is the same for both DNA and RNA, because both DNA and RNA has a hydroxyl group at 3' carbon. The only difference is DNA doesn't have hydroxyl group on the 2' carbon. So, and then basically in RNA, what you have left is this 2' hydroxyl group is still available for chemical reactions. But for DNA, there's nothing here. There's nothing on the 2' hydroxyl group. Okay. And another thing is this. So it looks like to form the phosphodiester bond, you only need the hydroxyl group here. But in reality is this. To add a new nucleotide to the growing hydroxyl group, what you need is not one phosphate group. You need 3 phosphate group. Okay, so this means to add like the third nucleotide, what you need, well, suppose this is DNA, what you need is DNTP, not DNMP. Okay, because although you only need one phosphate group to bridge this hydroxyl group with the next 3' hydroxyl group, but the incoming nucleotide always have 3 phosphate group. It is because you need energy to form this bond, and only the triphosphate nucleotide has enough energy. When this bond is broken, you can form the phosphodiester bond here. Okay, the incoming nucleotides always is NTP or DNTP. It's not NMP. Okay, that's another thing that's very important. Okay, so now amino acid use peptide bonds, and nucleotide uses 3' to 5' phosphodiester bond. And also, similar to proteins, in nucleotide we also use non-covalent bonds to stabilize high-order structures. And the non-covalent bond in DNAs or RNAs is the hydrogen bond. It's also first and the most important thing is hydrogen bond. Let's see how it formed. And, well, just to remind you, hydrogen bond is formed between, hydrogen bond is this. Whenever you have an oxygen or nitrogen or fluorine, but we don't have fluorine in biology, and then connected to one hydrogen, then this hydrogen is strongly positively charged. Well, it's not one complete charge, but it's strongly positive charge. And then there's another, either oxygen or nitrogen nearby. You would form attraction between this strong positive charge and this strong negative charge. And this is a hydrogen bond. Any hydrogen bond works like this. It's basically an electrostatic interaction between positive and negative charge. But any hydrogen bond takes 3 atoms, and 2 of the atoms has to be oxygen or nitrogen, and the one in the middle must be hydrogen. This is a hydrogen bond. And now when we look at the base, okay, this is the base for adenine, this is the base for thymine. And you can see that in the adenine we have an amino group which has nitrogen here, and it's connected to a hydrogen, like hydrogen here. And in the thymine there's an oxygen, so oxygen here. So you can form a hydrogen bond between the 3 atoms. And it just happens that the next one, in the adenine there's another nitrogen here, okay? And it's in a position so that in the thymine there's a nitrogen, there's again a nitrogen connected to a hydrogen, and this can also form hydrogen bond here, right? So if we can position A and T at this orientation, okay, hydrogen bond needs an orientation, then we can form 2 different hydrogen bonds in between, okay? This is for A and T. And for G and C, it's the same. So, and it's the chemical structure of G and C happens so that when we put G and C in this direction, so here's the carbonyl group here, it's an oxygen, and on C there's the amino group here. You can form a hydrogen bond like this. And in the middle you have a nitrogen, NH, and then there's another nitrogen here. You can form the second hydrogen bond. And then it has another amino group here, and there happen to be a carbonyl oxygen here, so you can form the 3rd hydrogen bond. So we say that A and T is a pair. It's a pair. A and T, and C and G can form a pair because their structure is just maybe by coincidence or maybe by luck. Their structure is complementary to each other, so that when they are positioned like this, you can form 2 or 3 hydrogen bonds in between. And another thing is, for A and T base pair, there are 2 hydrogen bond. For C and G base pair, there are 3. So the attraction between C and G is much stronger than A and T. Okay, this will become very important later. So this is what happens in DNA or RNA. So you have a chain, a line of growing nucleotide connected by 3' to 5' phosphodiester bond, and you can have another chain of polymers connected by phosphodiester bonds, and you can form a lot of hydrogen bonds in between. So this is kind of like the beta strands in DNA, in protein secondary structure. So you have 2 chains, and 2 chains can be attracted by hydrogen bonds. And also notice that the hydrogen bond it's always formed between one one is purine, the other side is pyrimidine. So purine has 2 rings, pyrimidine has only 1 ring. So it's always, if one side is a purine, the other side must be a pyrimidine. Otherwise, the pair would not happen. Okay, this is the first non-covalent bond in nucleotide. And the base pairing in DNA happens like this, because we will see that DNA always, most time, okay, in biology, DNA exists as 2 chains. DNA has 2 strands. So it's natural that one strand, the base on one strand would form hydrogen bond with the base on another strand. Okay, so DNA works like this. It has 2 strands, and the 2 strands are connected to each other by non-covalent bond known as hydrogen bonds. And it's different in RNA, okay? Because in RNA, in biology, in cells, RNA is made as a single strand form. Okay, DNA is double strand, RNA is only single strand. So in RNA, but in a single strand RNA, it also tries to form hydrogen bonds so that it can be stabilized. So what happens in RNA is it will twist, kind of like the alpha helix. It will twist itself so that the hydrogen bond can be formed. And so RNA, like say here, there is AU, GC, CG, AT. And so like this, it has a U here. It cannot form and with the U there is a C on the opposite side. It cannot form hydrogen bond with the C here, but so it can form hydrogen bond with the next A here. So for RNA, because it's a single strand, so this kind of works like the alpha helix. Although it's a single strand, it just itself can make all the twists and make all like just twist the strand around so that the maximum number of hydrogen bond is formed. So for the DNA, it's kind of boring. It's always like the 2 strands and form hydrogen bond in between. And for RNA, because it can twist itself, so RNA has a special shape. Okay, each RNA, depending on its sequence, it can have different shapes. And so this, again, this is kind of like the proteins, because if for proteins, different shapes can different primary structures can lead to different tertiary structures. And for RNA, it's the same. Different primary sequence can lead to different tertiary structures. Questions? No. Okay. And that means RNA, they are similar to protein in a way that they can form distinct shape, and RNA can be utilized to categorize. Okay, so RNA is, on one side, it's similar to DNA. It's composed of nucleotide. It has these AUCT letters. On the other side, because the single strand nature of RNA, it can form shapes like kind of like DNA. Okay, so that's a special thing about RNA. That's why people have a theory that before DNA and protein appeared on the Earth, there is a period on the Earth that's composed of completely RNA. That's called RNA world. There was a time we have the RNA world, because proteins is for doing all the functional stuff. DNA is for storing the information. And RNA can actually do both. So we think before DNA and protein appeared, everything could be done through RNA. Okay. And then there is a second stabilizing force. So for proteins, we have hydrogen bond hydrophobic interactions, van der Waals interactions, and salt bridge. And for nucleotide, we have a special stabilizing non-covalent bond, which is called base stacking. Okay, the base stacking it works like this. The stacking means, in chemistry, it means when we have two aromatic rings, okay, and there will be, and when the rings are positioned like this, there will be attractions between the two rings. Well, this happens because of this. Because in any aromatic ring like this, it's actually this. So on the ring, it's actually a lot of pi, it's called pi electrons. So it's actually the pi electrons. And when you have two rings like this, sorry for the bad drawing. So the next ring can also have a lot of pi electrons. And these electrons can interact with each other. And so basically, eventually what happens is these electrons, it forms a big electron cloud around these two rings. So this is called electron resonance. But the bottom line is when you have two aromatic rings, there will be attractions between the two rings. These are called base stacking. And so this is either DNA or RNA. So what happens is this is one base here, this is another base. So this is pyrimidine, this is purine. And besides the hydrogen bond in between, there is attractions between this base and the base above and the base in below. Okay, so this interaction basically, it keeps all the bases basically in this vertical space. Okay, so two types of, now we have two types of non-covalent bonds in the nucleic acids. One is called hydrogen bond, one is called base stacking. Okay, we have 4 different non-covalent bonds in proteins and 2 types of non-covalent bonds in DNAs. And again, non-covalent bonds are pretty weak, so we can denature proteins by using heat, right? By increasing the temperature. We can also denature nucleic acids by increasing the temperature, because the higher order structure of nucleic acids is also stabilized by non-covalent interactions. Okay, so that means we can denature both. Okay, and that will come handy later. Okay, and another thing is because we have the 3' to 5' diester bond, that means whenever we have an amino acid, we always have one end of the nucleic acid is 5', and the other end is a 3' hydroxyl group. So every strand, every DNA strand or RNA strand has a direction. One side is 5', one side is 3'. It's just like for proteins, we always have a direction. One side is N-terminal, one side is C-terminal. Because for any amino acid, there will be one side is amino group, one side is carboxyl group. And for DNA, it's the same. One end is the 5' hydroxyl group. The other side is 3' hydroxyl group. So there's always a direction, 5' and 3'. And to write a nucleic acid, the convention is this. The convention is always you write from 5' to 3'. Okay. So we can write this is well, it's hard to tell. Well, it has T here, so this is the DNA structure. So this DNA strand, it has a base ACGTA. Then you always write from 5' end to 3' end. So we write like this, like 5' ACTGA. So then we know A is always, the one on the left is on the 5', the one on the right is 3' end. Okay. And also just like proteins, if the peptide has somewhere less than 50 amino acids, we call them oligopeptides, okay? And the nucleotide, if it's less than 50 nucleotide, we call them oligonucleotide, okay? So this is just convention. By convention, all proteins are written from N-terminal to C-terminal. All DNA or RNA are written from 5' to 3', okay? And so this is about the structure of nucleotide and the nucleic acids. And now I want to talk about something a little bit different, because nucleotide can actually have many different functions. The first function is nucleotide are building blocks for nucleic acids. Okay, that's what we just talked about. Nucleotides, they are building blocks. They can form the polymer, which is then called nucleic acids. Nucleotides are also energy currency in cells. Does anyone know what's energy currency in cell? ATP, okay. And when we talk about ATP is, A means adenine, okay. ATP basically is one of the nucleotide for RNA, okay. And also nucleotide can be a precursor for a universal electron acceptor. We will talk about this. And nucleotide can be signaling molecules. Okay, first, ATP. So what is ATP? ATP looks like this, okay. It's exactly ATP. The RNA has 4 types of nucleotide, AUCG, okay. ATP, UTP, CTP, and GTP. And one of them, ATP, is used as the common energy currency. Okay, let's see what this actually means. This means this. When we want to make amino acid to proteins, okay, when we want to form peptide bonds and make proteins from all the amino acids, the delta G is very positive. And if you remember organic chemistry, when delta G is positive, that means the reaction would not happen automatically. When delta G is positive, that means you start from a low energy and now you want to go to a high energy, and this would not happen. Okay, this kind of reaction, it just would not happen. So, but we know amino acid does form proteins in cells. So how this happens is this. The ATP, this reaction from ATP to ADP, T means 3, D means 2. So when ATP hydrolyzes one of its phosphate group, okay, so when this bond gets broken, this will release a very large energy. So it's like the delta G is very, very negative. So it's kind of like this. And what happens in cells is we couple the two reactions together. Couple means like we add the two reactions together so that we make it so that when amino acid is it's forming proteins. At the same time, ATP is hydrolyzed. The coupling works like this. You have a reaction. You have a reaction that, for the reaction to complete, the energy is actually increased, and this would not happen spontaneously. However, you have another reaction, like ATP hydrolysis. This reaction, it will happen automatically. The energy will go down. And you let the two reactions happen. You design a chemical path, a chemical reaction path, so that the two reactions must happen at the same time. So as one reaction going down, and at the same time you will pull this reaction, this other reaction uphill. This is called coupling. This is called coupling. You couple a chemically impossible reaction to ATP hydrolysis. Couple means the two reactions must happen at the same time. Okay, for the amino acid forming protein, the ATP must be hydrolyzed, and the energy in the ATP hydrolysis can be used to driven the other reaction. This is called coupling. And in biology, almost all energy required reactions use ATP as the downhill reaction. So that's why this is, we say ATP is a universal energy currency, because whenever we have a reaction that's impossible to happen, we always couple that to ATP. That's why ATP is the energy currency. And by the way, so why we cannot use it? So if a reaction is not possible, can we use enzymes to make that possible? Like for amino acids, it cannot form proteins automatically. Can we use enzymes to make it happen? Enzymes speed up reactions. The answer is no, okay. Enzyme only speed up reactions, speed up possible reactions. Enzymes will not change the delta G here. This is the delta G, okay. The delta G between product and the substrate. The enzyme would not change this number. The enzyme only would let a reaction get to its equilibrium better, but the enzyme would never change the delta G here. If some reaction is not possible, even with enzyme or any catalyst, it would not be possible. Okay. And in the next chapter, we will talk about if enzyme cannot change delta G, then what enzyme can actually do. Okay. Okay, the second type of function carried out by nucleotide is called electron carriers. Okay. First, well, this is another chemical compound called FAD. Okay. It's called flavin adenine dinucleotide. Okay. We know what adenine is, so this part, this is a very big molecule, but let's just look at this part for now. Okay. This is an adenine. Okay. Right. This is adenine. This is ribose. This is phosphate. This is basically an AMP nucleotide. Okay. And then we have another part. We have also a 3-ring structure here, and connected to a 1, 2, 3, 4, 5 carbon. 5 carbon. Each carbon is connected to a hydroxyl group. 5 carbon here, and then we have another phosphate group here. So now this kind of looks like a nucleotide, right? Well, it looks like a nucleotide. It's not a real nucleotide, okay? But it looks like a nucleotide, and it just connected to the AMP by the phosphate bonds. Okay, so that's why we call them dinucleotide. Flavin. So this ring is called flavin because it has a yellow color. Now we have a dinucleotide. So a dinucleotide, it has on one ring it has a flavin ring. On another ring it has another nucleotide. It has a adenine. And so, well, by the way, this part is also called flavin mononucleotide. This part is called FMN. It's also a very important structure in cells. And this part, so this ring plus the 5 carbon, this one is also called riboflavin. this is basically vitamin B2. Okay, this part from the 3-ring plus this 5 carbon. This is vitamin B2. Does anyone know what a vitamin is? What's a vitamin? You heard that every day, but what is vitamin? Vitamin is chemicals that your body needs, however your body cannot make. Your body absolutely needs this, and we will see what its function is. Your body absolutely needs this, but we and probably other animals, they lose the ability to synthesize these chemicals in our own body, so we have to get it from diet, from food. This is vitamin, and all vitamins works like this. Your body needs this chemical, but it cannot make it. It can be made by plants, it can be made by bacteria. Well, it exists in other animals that also eat other bacteria or eat plants. So your body has to receive vitamin from food, from diet, because it cannot made by our animals anymore. Okay, so this is one type of vitamins. And the interesting thing, well, the function of this FDAD is this. It's involved in many redox reactions. Okay, its function is a redox. Do you know what redox means? Redox means means reducing. Ox means oxidative, oxidizing. So whenever you have a redox reaction, you like one part of the reaction give electrons to the other part of the reaction. So for the FAD, what happens is this nitrogen here and this nitrogen here, both nitrogen atoms can receive one electron, well actually 1 electron plus 1 hydrogen, and form this structure FADH2. And so this is basically half the redox reaction. Okay, we'll continue next class.